Hi, everybody, and welcome to today's webinar. Today, we are having actually a panel discussion. The topic, as we'll elaborate on, is translating from Arabidopsis research to other systems. We've got four great speakers who we'll introduce in a moment. Your hosts today are Adrienne Roeder from Cornell University and Cris Argueso from Colorado State, and they're both editors at the Plant Cell Journal. The focus issue is online. It's the May 2025 issue, and it was edited by Adrienne, Cris, Gabriela Auge, Xin Li, Lucia Strader, Cristobal Uauy, myself, and Shuang Wu. As I said, it's online now at the Plant Cell website. We also want to thank those who helped us with this beautiful cover. Quick message that we've just opened the call for assistant features editors. You scan the QR code or visit blog.aspb.org for more info. It's an excellent program for early career scientists to get a little experience in the publishing world. We'll be posting this video to YouTube. We want to make a thank you to our sponsor, the American Society of Plant Biologists. If you're not a member, you might want to join us, and you can use the code PRESENTS10 to get 10% off your membership dues. Finally, we would like to ask you to consider joining us at the plant biology meeting this July in Milwaukee, Wisconsin. And I have a note that the rates for registration will go up June 10th. So if you're going to join us, get busy and register now. Please put questions for the speakers into the questions box. Email me if you have any questions. If you're having trouble with audio, try checking your internet speed or dialing in. I know many of you need to receive a certificate of attendance. In 24 hours, you will get an email with information about how to request one. And now I'm going to turn it over to our hosts, Adrienne and Cris, who will take over.
Hello, everyone. Welcome. It's so great to have you here with us. We have a very exciting panel this morning talking about Arabidopsis and how it does or does not translate to crops and beyond. This is the subject of our focus issue that we've been really excited about putting together. So I'm going to kick it off, and then Cris and I will alternate, and I'd like to kick it off to the panelists by saying, please introduce yourself and tell us about what your lab studies very briefly and how you use Arabidopsis or Arabidopsis knowledge. And then please tell us what's your contribution to the translational focus issue. And so let's kick it to Anna to start it off.
All right. Greetings from North Carolina. I'm Anna Stepanova. I'm professor of plant biology and genetics at North Carolina State University. I study plant hormones, predominantly ethylene and auxin, as well as the interactions between the two. I also study gene expression regulation at translational and transcriptional level. I've been using Arabidopsis as my primary model for over 30 years. And as the funding climate in the United States has been gradually shifting towards more applied research, midway through this journey, I started also using other plants, primarily tomato and two Nicotiana species, Benthamiana and Tabacum. I continue using Arabidopsis as my primary system for most discovery-focused and proof-of-concept studies, and then move to other species as needed to test the translatability in the generalizability of our Arabidopsis-sourced findings. One of my additional interests is development of molecular tools and methods, and I can tell you there's no better system in plant biology than Arabidopsis to do just that. And specifically for this special issue, I collaborated with my talented graduate student, Anna Yushchenko, and my collaborator, my long-term colleague, Dr. Jose Alonso, to write a review article on Arabidopsis as a model for translational research. What you may possibly find curious about that paper is that the review manuscript started out as a preliminary exam for a PhD student in the lab, Anna Yushchenko. And I chose this topic for her as this was a primary theme of many discussions by the North American Arabidopsis Steering Committee, of which I was a member for 6 years, where, among other things, we brainstormed the best ways of making the broader scientific community, as well as the general public, aware of the role Arabidopsis played and continues to play in plant biology and beyond. My student Anna did an awesome job on her prelims, so Dr. Alonso and I went ahead and expanded that write-up into a more comprehensive manuscript for the plant cell. Okay, I'm ready to pass it on to Federico.
Thank you, Anna. I find a lot of similarities in my career. I did my PhD in Argentina. I'm Argentinian, in the role of transcription factors in nodulation in legumes. And then I moved to France. I joined Martin Crespi's lab in France to study long non-coding RNAs, for which Arabidopsis emerged as a perfect model because it was a simple model with many genetic tools for a complex question. It is how to understand and how to uncover the roles of the non-coding genome. Then I came back to Argentina. I set my own lab. We still work in the role of long non-coding RNAs in chromatin dynamics, especially genome topology, the 3D organization of the genetic information in the nucleus of the cell. And we always undertook fundamental research until we understood actually how we could use our understanding of RNA biology to develop biotechnology for sustainable agriculture. In 2023, we published in Nembo Journal a work by our talented PhD student, Florencia Mamarella, where we sprayed an epigenetically active long non-coding RNA called Apollo to Arabidopsis plants, and we modulated auxin homeostasis and the response of the plant to the environment, particularly jade avoidance syndrome. Based on this breakthrough, we set the basis for the development of RNA carriers, and we started developing RNA sprays. And then we launched a startup company that is called after our favorite RNA, Apollo Biotech. And we are developing RNA tech for agriculture, both to replace synthetic pesticides by a sort of RNA vaccines, but also we are developing RNAs to acclimate plants, crops, to environmental constraints such as temperature extremes, drought, and some other abiotic stress. So Arabidopsis was a key resource for us. It is still for us the best model for fundamental research. We can use or combine several genetic tools to develop our research. And then we included more crops, model crops such as tomato, as Anna, because it has other characteristics that are closer to bigger plants, let's say. But we still work in Arabidopsis, we will still do it.
Let's pass it to Xiaolan, maybe.
Okay, hello everyone. So my name is Xiaolan Zhang and from China Agricultural University. So I got my PhD from University of Georgia and my working on the maize, especially the maize leaf development. After that, I did my postdoc at California Institute of Technology. At that time, I studied about the Arabidopsis, about the flower development. I come back in 2011. So since then, I switched to cucumber. So I work on cucumber, especially the fruit development and also the shoot branching. So especially about so basically I use those many of the knowledge from the Arabidopsis and also from the corn to explore basically the area in the cucumber. So many of the fruit shape genes and also the shoot branching and the regulatory networks. And also I was trying to develop a new molecular tools so that to help the cucumber breeding. So especially to get a very good appearance trait and also to have a very good shoot architecture. And I already have 3 cucumber germplasm. It's based on my cucumber molecular markers to help you do that. And in these issues, I contribute to one review paper. It's together with other colleagues. It's about the developmental pathways in plants, and especially the lessons from Arabidopsis for crop innovation. And also I contribute to a research article. It's about the SMP change in the CEP2
contribute to the fruit length variation in cucurbits. Yeah, so that's all. Thank you. And switch to next.
Thanks. Mike?
Oh, hi. My name's Mike Bevan. I'm an emeritus fellow at the John Innes Centre. My current research involves trying to understand the epigenetic consequences of hexaploid wheat formation. This involves studying crosses between diploid goatgrass, Egalops tauschii, and tetraploid durum wheat. And what we're interested in is how, when you add these three giant genomes together, what are the consequences epigenetically? The short answer is that there are profound changes that affect the compaction of chromosomes. So in a diploid, in the diploid grass, the chromosomes are extended, and when they come together in a hexaploid context, they are substantially compacted. This compaction leads to reduced chromatin accessibility and reduced overall transcription. And this has got us interested in what are the mechanisms that control this sort of thing, and they're very, very poorly understood, even in animal systems. So at the moment, we are just writing this work up, but in order to get at the mechanisms in wheat requires a lot of important fundamental work in Arabidopsis, for example. But whether things happen at this scale in Arabidopsis, what happens in wheat happens in Arabidopsis is not at all clear. My other project focuses entirely on Arabidopsis, where we are working to understand the function of a set of genes that encode peptidases. These peptidases are regulated by phosphorylation, mediated by multiple regulatory like kinases, such as BRY1, TMK1, TMK4, clavata3. All of these things negatively regulate the activity of this peptidase. And what we think it does, well, we know it cleaves a whole lot of regulatory proteins. So we think it's DAR1 is part of a system that remodels the proteome in response to hormone and peptide signals, such as clavata1, clavata3 system. So that system, the DAR1 system, we've tried, and other people have tried to see if its effect on size control has an influence on, it has a big influence on seed size and yield in Arabidopsis, but that does not translate into crop plants as far as we've worked. Strangely enough, what happens is the DAR1 mutation in poplar trees makes the poplar cambium undergo additional division, so you get a bigger tree. Who knew? That's part of the excitement of Arabidopsis research. So historically, I was involved in the initiation and completion of the Arabidopsis genome project, which is 25 years ago, and since then I've been involved in a variety of other plant genomic projects, most recently the wheat genome project, where we did an initial survey sequence. My contribution to this plant cell special issue, my pal Cristobal Uauy asked me to write about the links between Arabidopsis and crop species, how genomics links them, and also about some of the more social / political aspects of getting a big genome project off the ground, and how to bring the community on or not, and how this affected the balance of Arabidopsis and crop research, which I think it's a lovely balance at the moment, but in the past there were wouldn't say there were misunderstandings, but there were certainly a lot of issues that needed to be resolved to make sure that Arabidopsis has its fully deserved place in the heart of plant science.
Thank you. I'll pass it to Cris for the next question.
Okay, so yeah, I just want to introduce myself really quickly, Adrienne. I think you should do that when you go for the next question. I'm Cris Argueso. I'm an associate professor at Colorado State University in Fort Collins, Colorado. It's kind of cold here, so I have my fireplace going on. It will get warmer later in the day, but the early mornings in Colorado, even in the spring, can be pretty cold. It's a pleasure to have all of you here, the people that are attending the webinar. Also, thank you very much for our panelists. And I work on plant hormones like Anna. I'm very interested in the small molecules that regulate plant development and responses to the environment. All right, that's enough about me. I'll just go for our questions so we have time to discuss science and talk about how amazing Arabidopsis has been and useful to all of us. So my question, the first question that I'm asking is, so this year, it marks actually 25 years that the Arabidopsis genome was sequenced. It was the first plant genome to be sequenced, one of the first few eukaryote genomes to be sequenced. My question is, how has this helped plant biology in general, in fact, biology in general as well, in your opinion? So to our panelists, how do you think the sequencing of the Arabidopsis genome has helped plant biology and biology in general?
I'm happy to take this. Undoubtedly, I think everybody in the audience would agree that having the genome sequence available have helped us tremendously. If you think about cloning genes in the past, for example, to identify causal variants behind interesting mutations, that used to be a multi-year endeavor. And these days it takes weeks, if not days. And having a reference genome and genome annotations available to us gave us a list of candidate loci to focus our efforts on, so that the time can be spent on characterizing the gene function rather than trying to clone a gene. And when I was a grad student in the late 90s, that is before the genome sequence was released, even things like figuring out whether a cDNA clone for a gene of interest you wanted to overexpress was or was not full length was super tricky. Now, with a good reference genome, ample RNA sequencing data, and accordingly strong experimentally validated annotations, we know exactly what part of the gene is being transcribed, if there are any alternative splicing products, and if we have RiboSeq for the data set, we also know with high confidence where translation starts and ends. All of that would not have been possible without a reference genome. So definitely, I would say every scientist benefited from having a reference genome available.
Yeah, if I may add and comment on that, you know, we knew when we started sequencing the Arabidopsis genome that we would remove a whole lot of work. Physical mapping was expensive, time-consuming. Most of you have probably never had to do it. But one thing that I found and my colleagues found very surprising and very pleasing about the impact of the Arabidopsis genome is it really democratized plant science. What I mean by that is even small labs, anybody with a good idea could go to the genome sequence, could go to the stock centers, because remember, as part of the Arabidopsis genome project, there were parallel projects. I was also involved in setting up stock centers, the ABRC in Ohio and the Nottingham Arabidopsis Stock Center. So in parallel with the genome project was the formation of collection and standardization and distribution of genetic resources to anybody. So this made doing research on Arabidopsis much easier for a larger number of people. So you get more people into the field, you get a far wider range of activities and capabilities and things like that. So it's the social context, I think, has been one of the biggest and most important contributions of the Arabidopsis genome project. And it also helped focus the community on working on one particular organism. Prior to Arabidopsis, the main genetic systems were maize, which is, of course, still a brilliant system to work on. It can answer many questions that are perhaps you can't in Arabidopsis. But it required, you had to work at a special university, Cornell, Columbia, or something like that, in order to be able to do maize research at Iowa and such like. So now you can work in a small university. All you need is a growth cabinet and the internet, and you're off in Arabidopsis research. And it took away a whole lot of research that was scattered about a whole lot of different plants and model systems, petunia, tobacco, Nicotiana plumbaginifolia, all contended as potential model organisms. And this meant that if you discovered something, bringing it together in a unified whole was not possible. So Arabidopsis genome tended to unify research for a larger number of people, more ideas focused on one plant. That for me has been perhaps the most surprising outcome of the genome project.
Yeah, thanks. Aligned with what has just been said, there was also the possibility with this first plant genome to compare plant genes with mammalian genes, and also Arabidopsis set the basis for the analysis of gene ontology in plants and phylogenetics. It accelerated the way we could classify genes, organize genes in families. There were many genes that were plant specific, and we didn't have a clue about that until we sequenced and we started analyzing the Arabidopsis genome. So I think it was clearly a breakthrough that helped to develop plant genomics in general.
Awesome. Thank you all. So as Cris pointed out, I should introduce myself. I'm Adrienne Roeder. I'm a professor at Cornell University, and I work primarily on Arabidopsis, and then we add in some other fun things like moss occasionally, diatoms, algae, things we think are fun. My lab studies plant growth and development, how organs reach the right size and shape. Okay, so our next question for the panelists is, what are some of the most successful examples where knowledge from Arabidopsis has directly led to improvement in crop species? And maybe Xiaolan, you want to take that one?
Yeah, sure. So because my major is vegetable science right now, so I would like to give 2 examples about the vegetable crops. So one is the lettuce. So the lettuce, the flowering, if they got bolting, there will be a huge damage to those products. But about what kind of thing to control the bolting, it's completely unknown. And the lettuce genome is very complex. So we based on the knowledge from the Arabidopsis to control the lettuce and also the cabbage, Chinese cabbage, the flowering pathways found it's all conserved, especially like the vernalization pathway, and also the temperature controls that. Like the lettuce is the high temperature to control to make them bolting. But the Chinese cabbage, they need the low temperature to induce the bolting. So we think the knowledge from the Arabidopsis, the flowering pathway, is very, very important to make us to understand, and also to the industry to control, to make the technologies to what kind. So that we, so that we know it's which techniques we could prevent their bolting or flowering in these crops. So yeah, we think that's very, very important. Yeah, thanks.
I can provide a couple of additional examples. I think many of you are aware of Arabidopsis morphogenesis genes, such as baby boom, bushel, and their crop orthologs. These genes made the transformation of recalcitrant crops feasible. ectopic expression of these developmental regulators makes the formation of somatic embryos possible, which enables development, makes possible genotype-independent regeneration of plants. Another great example you can find in the literature is the manipulation of fruit size, such as in tomato and physalis, by introducing cis-regulatory mutations and deletions in the promoters of genes of the clavata and Wuschel signaling pathway to downregulate their expression, and mutations that generate enlarged meristems and lead to flowers with extra organs ultimately result in this multilocular, bigger fruits that are more appealing to consumers. And I want to also cite a couple of recent examples from a private company in North Carolina, Pairwise, where glucosinolate-free mustard greens, as well as seedless blackberries were created. And it is the actually functional annotation from Arabidopsis of genes that provided these researchers with creative ways to identify candidate genes to target via genome editing.
For me, one of the first and most important examples of Arabidopsis guiding the discovery of a gene in a crop plant was Nick Harbert's work, where he had mapped a gibberellin-insensitive dwarf mutant in Arabidopsis and found the GAI gene, part of a transcriptional repressor complex. And a lot of people, of course, dominant dwarfing genes have been essential in the domestication of modern wheat. And we had at the John Innes at that time, there's a lot of work going on in wheat, a lot of work going on in Arabidopsis, and there's a gulf between these two. So what is the point of working on this model system? It's most important that you work on crops. That was the tenor of the discussions that we were having, until Nick Harbord and Mike Gale found the same gene in wheat and the same range of allelic variation in that gene that gave rise to wheat with different heights. And then the light bulb went off in this quite conservative crop science community, and bingo, one of the links between Arabidopsis and wheat, which I, in my estimation, is quite a long link to make, was successfully made. So that's the example that's pretty dear to my heart.
All right. Okay, so we'll go to our next question here. So if you guys haven't seen the focus issue, it's an amazing focus issue with many research articles as well as reviews. And one of the reviews talks about how some things just don't translate super well from Arabidopsis to crops. And while this can be disappointing here and there, it can also be very illuminating. It can let us know about different mechanisms in the different plants. So the question that I have for the panelists is, how do the genomic and physiological differences between Arabidopsis and crops complicate the translation of findings?
I would like to take this one, especially concerning what we are doing in our lab and our company lab, that is the use of the potential of RNAs to develop technology. That is actually what we contributed to with our review article. And Arabidopsis is a very particular species because it holds a very compact genome. The non-coding genome ratio is pretty low compared to other species. Arabidopsis genome is about 40% non-coding or something like that, whereas perhaps between 50 and 60, whereas, for example, wheat is over 90. So that is a key point if we are studying how the non-coding genome and non-coding RNAs are participating in how chromatin dynamics respond to the environment. And especially if we are going to develop RNA-based technologies to modulate this and try to trigger responses in a plant. So this is a main issue. I think that to this end, tomato is a very good species because the size of the genome represents a wide range of plant species and crop species. And there is another thing at the physiological level, that is that when you're working with RNA splice, Arabidopsis is very sensitive. is very fragile. So that makes it very difficult to work with because the plant is stressed as soon as you spray RNA on it. So again, tomato or other crops, legume crops, for example, are very good models to develop RNA-based technologies instead of Arabidopsis. Still, we are using Arabidopsis to better understand the molecular basis of exogenous RNA sprays because of the genetic tools. We can use specific mutants of the small RNA biogenesis pathway or whatever to assess how these RNAs are absorbed and processed into defense molecules in particular.
Okay, and also I want to add 2 more points, 2 more examples, like the cucumber, the fruit, and compared to the Arabidopsis fruit, because the Arabidopsis fruit, they need to open so that they could dispose their seeds, and all those genes control the fruit opening genes, but they do also function in the fruit development in cucumber. But the cucumber fruit, they never opening. So all their function has been diversified, and such as the fruit fall, it controls the fruit length. And also like all these transmitting tract genes, they function on the other things of these, like the shape and also the fruit, the trichome density, all these things is diversified in the cucumber system. And another example is the Chinese cabbage. Although many of the genes in the Arabidopsis working on the leaf development, the function is conserved. But there is one special thing about the cabbage, because they have a heading, like the leaves were curved together to make a heading, and all those genes is completely new, because the Arabidopsis, their leaves, they were not form this heading. So all these kind of things, but it's very important for the like the Chinese cabbage, their industry. But yeah, the knowledge from the Arabidopsis, they cannot directly translate to this system. Yeah.
Yeah, I would add that there's no substitute for a good or a strong fundamental understanding of crop like a cucumber or wheat or tomato, understanding its physiology and growth and development. Of course, those are completely different from the little Brassica Arabidopsis. But when you get down to it, from what we know about molecular mechanisms, those molecular mechanisms can be a very good guide or shortcut, that it's not necessarily exactly the same. The gene doesn't execute exactly the same developmental or environmental function in Arabidopsis and in a crop plant, but its basic mechanisms are often the same. Hence, I think that when people have followed up candidate genes in crop species that have a particular phenotype in Arabidopsis, and they expect a similar range of phenotypes in a crop plant, then you can be disappointed unless you know your physiology and development in the crop plant very well. And what I would add, and just sort of tried to make clear in the article we wrote for the special issue, is that the more genomes you get and the more pangenomes you get, you've got an incredibly rich resource that is sort of stepping stones across evolutionary space that gives strength to your interpretations of gene function from Arabidopsis to crops. So I think there'll be less and less disappointment.
Yeah, I can also chime in here. While you can study certain processes, maybe climacteric fruit ripening or nodulation in Arabidopsis, because Arabidopsis doesn't have climacteric fruits and doesn't make nodules, you probably can study perennial traits or C4 photosynthesis in Arabidopsis because it's an annual C3 plant. But it's important to realize that even the study of this divergent processes in crops still benefits from the store of Arabidopsis source knowledge. For example, I'm an expert in ethylene signaling, and apparently, I don't know if you've heard, like in a commercial tomato breeding, a never ripe trait in tomato is commonly used. And NR, this never ripe trait, is actually one of the ethylene receptors. So understanding why tomatoes never ripen and the physiology behind that trait comes from Arabidopsis receptor mutants. So now this NR trait is widely used in commercial hybrid tomato production because NR heterozygotes are firmer, amenable to long distance transport, have an extended shelf life and flavor retention. And a lot of what we understand about this NR trait comes from Arabidopsis studies.
Yeah, there's many examples of that. And the more we understand about basic fundamental mechanisms in Arabidopsis and integrate these together, like you mentioned, Anna, about integrating auxin and the ethylene pathway, and understanding the ethylene pathway more deeply, all of this knowledge will have gives more and more power to the applications in diverse plants.
All right, so I pass it to Adrienne now.
It's always fun to talk to my non-plant colleagues, and they all know auxin from the auxin-degron system. So that brings up the next question, which is the impact of Arabidopsis research goes beyond plant science. Would any of you like to elaborate on some of these broader impacts?
I can start. Go ahead.
No, thanks. I'm just about to, blow a trumpet for my colleague Caroline Dean, whose basic work on chromatin structure regulation, environmental regulation of chromatin structure, basic mechanisms of transcriptional control, which is fundamentally important in all eukaryotes. Arabidopsis has had a very important role in understanding some of the basic mechanisms of transcriptional control. And I'd add my colleague Yiliang Ding's work on RNA secondary structure as well. That is a compelling example of Arabidopsis just providing a fantastic experimental system for understanding a universal process.
Yes, another very good example of this is, for example, the discovery of IPS1 induced by phosphate deservation 1, the long non-coding RNA described in Javier Pasares' lab in Spain that acts as a microRNA target mimicry that kidnaps the microRNAs, that was later discovered as a general mechanism of RNA sponge of microRNAs across organisms that, for example, circular RNAs in mammals. So many things were first discovered in Arabidopsis and showed to be true in other organisms, including animals. From my viewpoint, what I really like about plants in general and Arabidopsis in particular, when compared, for example, with mammalian cells or cell cultures, is how we can study the role of a particular gene, for example, a transcription factor, at the organismal level. No, instead of using cells to knock out, knock down, or overexpress a particular transcription factor and analyze the molecular output of this deregulation. In Arabidopsis, we can have a mutant and still have a living organism and try to understand what is the impact at the developmental level. And I think that is really complementary to what has been first observed also in animals for families of proteins that are conserved across kingdoms.
And I'd like to mention a few Arabidopsis-inspired molecular methods and tools. Intact, TRAP, DAPSeq, TILling, they were all first developed in Arabidopsis. And things like that Adrienne just mentioned, auxin-inducible degron AID, several of the optogenetic systems such as Cry2, SIB1, or 5B-PIF, as well as ABA chemically induced proximity systems, they all leverage Arabidopsis proteins to enable inducible expression of traits of interest. And they have been widely adopted in mammals and other animal species. So Arabidopsis is to thank for that.
All right, so if none of the panelists have any other input on this one, I'll switch to the next question. And in this focus issue, we had a commentary, and it was actually one of my favorite parts of the focus issue. And this commentary was about defense of plant science funding, not only Arabidopsis, but foundational plant sciences, not only in the United States, but all across the world. And I really recommend people to read it. And this is based on the idea that humanity is facing a lot of challenges. These are societal challenges with environmental degradation, food security, and of course, climate change. And then at this point, plant sciences becomes a really important topic of research that could have a direct impact on these challenges. So my question to the panelists is, what do you think is the role of Arabidopsis in these societal challenges that we are facing that are so important? And in the ability of translating knowledge from Arabidopsis to crops or other systems that could help with the societal challenges?
Let me take that. I think a large chunk of proof of concept science should still be done in Arabidopsis rather than in crops. This would be the good way to save both time and money. Arabidopsis is a workhorse that prior generations of scientists worked very hard on to equip it with tools and capabilities that facilitate further innovations and that can ultimately be moved to crops and beyond. But it would not make sense to abandon an Arabidopsis as a platform for plant science research, as it still has a ton of potential to continue to move the sciences forward at a faster pace and at a lower cost that any other flowering plant species can.
I'd follow up from that. It's what we don't know and don't understand. That is, in terms of mechanisms, environmental responses and such like, that we urgently need to understand to address, the problems of food security, which historically has always been the biggest problem that humanity has always faced. Getting enough food in the right place at the right time. And with global climate change, this problem is imminent and foreseeable. Now, what I mean by what we don't know is that in the review we wrote, we looked at, what is known about the however many 25 odd thousand Arabidopsis genes, and only about 40% of them have been touched in any way by any experiment anywhere in the world, as far as we can tell from the TARE website. Now, this means that there's a huge amount more to be discovered. And without this basic knowledge, which the best place to generate that and integrate it is in Arabidopsis, our abilities to understand how crops respond to the environment is going to be very, very limited, because in order to make new crops, you've got to understand, identify and partially understand which genes, which genetic variation is the important set of variation for environmental responses. I'll just give you one little example. It's a big example. And that is the process of meiosis is very temperature sensitive in some grasses, including wheat. You can get a major failure of the wheat crop. It happened in Europe in 1975, 76, when a new variety called Moulin was introduced. And unfortunately, it had a temperature sensitive step in meiosis, and crop yields went way down in a hot summer. So meiosis happens at a period in late May, early June, around about this time of the year. And if it's too hot, meiosis doesn't work. So people like Raphael Mercier and Graham Moore and others have now really starting to get to grips with the molecular mechanisms of meiosis, identifying the genes, identifying temperature-sensitive steps of meiosis. And with meiosis being quite a conserved mechanism, you'll be able to identify particular allelic variation in crop plants that might give greater fertility and increasing temperatures during key stages of growth. That's just one example, but there are dozens of others. So I think Arabidopsis, to answer the question more specifically, we've only just started to understand processes in plants. We've only got a very, very vague knowledge of how gene function converts is expressed in terms of cell behavior, cell interactions, orthogenesis, and all of that sort of thing. You know, Adrienne knows all about these sorts of things.
Yeah, and also I would like to share one more example related to the food security. So as the people become more and more pay attention to the food, whether the healthy food, and also like many of the functional components, such as the vitamin C, carotenoid, and also folic acid, and many of these important things for our human health. And many of these synthetic pathways, its knowledge is from the Arabidopsis. But like in the rice, also like in cucumber, like tomato, we could make many of these with many different colors, so that they also have many of these functional components. It's good for our health. So we think that those also we based on the knowledge from the Arabidopsis to make it much easier. Yeah.
So it's now time to turn toward questions from the audience. And there's one that I'd like to ask the panelists. And it continues our theme of looking toward the future. It says, it is great to hear of the past accomplishments of the model, but I'm interested in what the future can bring. Can anyone talk about what are the frontiers now in Arabidopsis research? What would the community try to tackle from this point?
Well, yeah, go ahead, Anna.
I was going to suggest taking a path through synthetic biology types of applications where you can leverage the high throughput cloning methods that synthetic biologists in yeast and E. coli now in plants developed, and then move this amazing tools to Arabidopsis. So synthetic biology has been lagging behind in plant sciences relative to other model organisms focused research. I suggest that we can take a more comprehensive approach. And for example, if there's a particular pathway that we want to bring to crops, let's say a particular drug that we want to produce in a certain species, in tobacco, let's say, right? So we can make certain precursors in yeast, but plants have a much more elaborate systems to make secondary metabolites. So maybe plants would be better suited to produce certain chemical compounds than yeast or mammalian cells or insect cells can deliver. So from the synthetic biology perspective, I think Arabidopsis and cell cultures of other species, including crops, hold potential. So that's one thing I'm very excited about. And also implementing gene regulatory circuits such as logic gates. I think Arabidopsis is a great model for that in plants.
I wanted to add something about about something that happened more recently. That is how the Arabidopsis community was pioneer in natural variation studies. Arabidopsis was a model for this, and I'm still curious about how this will evolve. Not only because of the resequencing or the 1001 genomes and the resequencing by long reads of a growing number of ecotypes, but also now we can start adding, for example, epigenomics to this layer of first genomics, and this will open wide perspectives. How plants adapt to the environment in having similar genomes, but perhaps alternative epigenomes. And in this sense, I'm also very curious about all the potential that holds single cell sequencing, but not only for transcriptomics, but also for epigenomics, that will be for sure move very fast in Arabidopsis compared to other plant species.
I would like to make a pitch for an area of research that everybody is interested in, that is the mechanisms of growth and development, and how mechanisms that are expressed in an individual cell actually emanate into the growth and development of an individual cell in its organ context, i.e. that involves cell-to-cell communication and how the final sizes and shapes of plant organs are formed. And this is a very fundamental question. It's one that we have a very, very sparse set of knowledge on. And this requires tools to be advanced. One of the tools is continued advance of light microscopy. You could see the great advantage of the transparent Arabidopsis root and how that has really strengthened our understanding about the formation of different cell types and different tissues and how they grow. But that information is lacking in most other plant organs. So improved microscopy, but most importantly, improved measurements of cells. Orthographics, for example, is a tool that is only used in a few laboratories. Adrienne's, for example, and Richard Smith at the John Innes and Miltos in Cologne. These labs are making tremendous strides in understanding the cellular basis of growth and development. And attached to morphographics or integrated into it is a modeling, because I think without there is so much information that's available now about basic processes, how the phenotype is derived from these mechanisms, the action of these mechanisms, is very poorly understood, and you'll possibly only get a hint of what's actually happening if you have models that you can test. So this is a move away from the old sort of descriptive biology and biochemistry into a much wider, richer scenario in which you can test different hypotheses about the integration of gene action and how those actions give rise to a cell behavior and organ behavior. That, for me, is a major frontier.
And perhaps integrating AI in the analysis of the amazing data sets that we've already accumulated in Arabidopsis, right? There's no other plant species that have as much data already available. So finding patterns that are not noticeable to the naked human eye now will be possible with the artificial intelligence and machine learning algorithms. Yeah.
Machine learning has got a long, quite a long history in classifying problems, classifying components, genomic components in particular. I would sort of, there's a lot of excitement about AI, but I really think that models that are based on a person's intuition about the biological process, rather than take all the data and put it together and, oh, look at this correlates with that. That's exciting. This is exciting. I think that way many rabbit holes lie. That I think there's no substitute for a finely tuned biological mind.
I'd like to mention one of the comments from the questions. It's actually a comment, just to follow up on this whole idea of the future. Gabriela Auge says, I'd like to mention that Arabidopsis thaliana has been a valuable tool to study the ecology and evolutionary biology of plants, especially the identification of genes and molecular mechanisms underlying adaptive traits. Thanks to the accessions collection, we can better understand the role of plants in natural ecosystems. And taking this point a bit further, it helps us to forecast responses in the face of climate change. We just need to do better linking molecular biology to eco-evo knowledge. So I thought that's also a comment that really fits well in this. I'll turn it over to Cris for the next question.
Okay, so we're gonna go with a question that was sent to us in advance to this webinar. And the question is, it's an interesting question. It says, will there ever come a time where Arabidopsis research has run its course, making it necessary to focus entirely on fundamental research in crop plants only? What do you guys, panelists, think of this question?
Never.
Not for a long time, I would say.
No one is biased here, don't worry.
Even once we know what each individual gene's role is, the interactions between different genes and molecular pathways, developmental processes, environmental responses, and so on, would be still much more efficient to study in Arabidopsis. So I think Arabidopsis will stay relevant for hundreds of years. That's my personal opinion.
I fully agree. The amount that we don't know is huge. And when you've got a genome sequence, right, you sort of get a rough estimate. You know, we know about this gene, we know about this one, and so far, it's a very sparse knowledge scape. And the genome allows you to sort of set a limit on what you might know. You know, the limit is set by the number of relationships and identities of genes. So I would say Arabidopsis is to coin a phrase, an endless frontier of discovery. It'll always be needed. It'll always be strong and central.
In every research project, it's always a key to choose the accurate model for the question we ask, no, for the hypothesis we have. And many times it's better to choose a simple model for a complex question, and also to choose a model where we know a lot about to find the question to specific, to find the answer to a specific question. So the more we know about Arabidopsis makes it more suitable for very complex questions, I find. So because if you move to another model where you don't know many things, there will be a lot of variables that you cannot regulate or control. So I think that is the more about Arabidopsis, it makes it more appealing for specific research.
Yeah, and also I think it's I agree. Like, to answer these questions, Arabidopsis is going to be much easier and also much efficient than other crops, and to not only like because the system is so mature, and also like if you change to another crops to answer this question, you need much more space to grow the plant, and also much more time and effort to establish the system. Yeah.
So I guess that means that we're going to be in business for a long time. That's a good, good take-home message from this discussion.
Mm-hmm.
So I'd like to close out the webinar with one last question. And it's from an anonymous attendee. It says, would you recommend that students and postdocs cross-train in Arabidopsis and another plant, e.g. a crop, so that they are best prepared for current future careers, or is it sufficient to focus on a crop only?
I think having multiple skills is always better than a unique skill, because the job market is unpredictable. So if you have an opportunity to be trained both in a model organism and a more applied system, you're much more sought after as a candidate for a broader number of positions. So if you have that opportunity, yes, definitely jump on it and learn both systems. That's my take.
I think for the the question is for students and the postdocs, right? So I think if you, because you don't have that much time to be very good on both systems. So I think you could focus on one region or one area. You got very good publications and the knowledge on that one. And then if you find a job, then you are going to begin your own career, then you could change to another crops. So that's my opinion. Thanks.
I would say that working in Arabidopsis enables you to frame a question that is tractable. That is, you're sitting down, you're starting your PhD, you're given a field of research to work on. I think Arabidopsis enables a graduate student or a postdoc to be able to focus and to create, just looking at genome sequencing and relevant publications and databases, and then make frame hypotheses that can be tested quite quickly, relatively quickly for a plant using Arabidopsis. I think, you know, because grants are always too short, or rather never long enough, Arabidopsis gives you the power to try to answer ambitious questions as quickly as possible. So I would say to the person who asked the question, focus on making or framing good questions that you can answer in a feasible way. And if you need to learn about tomato or wheat or something like that, pick it up on the way. But it's the biological question that's the key thing. And Arabidopsis is good for framing those questions.
I guess that this always also depends on the organization of the research lab. I mean, science and research is a joint activity in a group with joint goals. And sometimes it's good for a PhD student to have a project in Arabidopsis and to contribute to long-term projects where we have questions that take time. For example, in crops, it will certainly take time. But then we can contribute to that, learn more skills, develop more skills in crops, but at the same time have, as Michael said, very sharp questions with a good frame in a very optimal model organism as Arabidopsis. I think that combination, as Anna also mentioned, that combination of both things is the best way to face a postdoc project or a PhD thesis.
Excellent. I'd really like to close out this webinar because we're at the hour, just after the hour. I want to thank our panelists for their super insightful comments and really interesting thoughts and discussion. I'd like to thank our audience for attending and for their really interesting questions. I'm sorry we couldn't get through all of them. Let's continue Arabidopsis into the future, I think is together with crops. It's really a advantageous system.
And I'd like to thank Cris and Adrienne for putting together this excellent panel discussion, as well as the editors of the focus issue. It's really great. It's a great read, a lot of interesting stuff. And I'm also going to plug, I put a link in the chat. The next journal webinar we have is from Plant Physiology. It's July 17th. It actually has some of the same authors as we have in our Arabidopsis focus issue. It's on numeracy, realism, and relevance in plant science. And it will also be in the panel discussion format. So that's going to be very interesting. July 17th, look at the blog posts. Thank you again for supporting our journals. We really appreciate it. Again, thank you speakers. Thank you editors. Thank you so much, Cris and Adrienne. This was one of the best hours I've spent recently. Thanks a lot.
Yeah, thanks,
Bye-bye.
Cris and Adrienne. Nice to meet you.
Bye. Thanks, everyone.
Bye.
Thank you so much.